Okay, this is podcast number 4 on Chapter 9, the nervous system. In this podcast, we're going to take a look at how an impulse is actually sent down the neuron. Anytime we send a nerve impulse, it is an electrochemical process. The chemical portion comes from the neurotransmitters that are sent through the synapse. The electro part of it is actually because we use two different ions, potassium and sodium, which are both positive ions. And we also need energy in order to make this process happen. What is basically going to cause an impulse to move down is that potassium and sodium ions are going to be moved across the cell membrane, and that's going to change the charge of the membrane. So this kind of works in a similar way to electricity flowing through a wire to a light bulb. Okay. So in a nerve impulse, anytime that we are starting our process, a cell starts at resting. So the resting potential of a neuron is when that neuron is what we call polarized. A polarized neuron has positive charges on the outside and negative charges on the inside. So there's a lot more positive ions outside than there are inside. This happens because of the process of, or the actions of the sodium-potassium pump. The sodium-potassium pump actually requires energy, and it ensures that sodium stays outside of the cell, and that you end up with more of these positive sodium ions outside of the cell than you have the positive potassium ions inside. So what you end up with is a net of more positive outside than inside, so that makes the outside positive, and the inside of the cell becomes more negative. So it's a relationship to each other. So at resting, we are positive on the outside, negative on the inside. Now, as a cell gets stimulated, which is going to start the action potential, we are now going to be switching these charges. So a stimulus to the resting neuron, say a sensory input from the outside, you're feeling cold or neurotransmitters are sent, whatever happens, this is going to open up sodium channels, and that's going to allow sodium to rush into the cell. When sodium rushes into the cell, it's going to temporarily depolarize the cell. That means that we're going to end up with the inside of the cell being more positive and the outside being more negative. Okay, so when sodium rushes in, we now are going to end up with more positives on the inside than the outside. At the same time that this is going on, the sodium's rushing in, potassium will start to leak out because we want to kind of get us back into the resting state eventually. But the action potential itself is sodium rushing in, and that's going to temporarily make the inside more positive and the outside more negative. From there, we have to get the cell back to its resting state. It doesn't really like to be in the depolarized state. It wants to go back to being polarized. So we have to get the membrane back to resting potential. So that means we have to repolarize the membrane. This means we're going to get the sodium back to the outside of the membrane and the potassium back to the inside. This requires the sodium-potassium pump again. It has got to use ATP because these guys don't necessarily want to go where we need them to go. So potassium is going to get pumped back into the cell because it's been leaking out for a while, and sodium is going to be moving back out. As we do that, we're going to go back to resting. Now, no action potential can happen again on that neuron until we have returned back to resting potential. So this step has to occur. We have to get back to resting before action potential can start again. Now, keep in mind, there's a lot of steps going on here, but this actually happens very, very quickly. It takes, a millisecond for an impulse to be sent. So what I'm going to do now is just kind of break these down into smaller steps. So number 1, we have our resting potential. So remember, resting potential, we are positive on the outside of the cell and negative on the inside. Okay, and that happens because there's more sodium ions outside than there are potassium. So there's more positives outside than inside. The action potential is what we're seeing here in step 2. So in the action potential, sodium ions are going to rush in, and that's going to make the inside more positive just at that spot, and the outside is going to be more negative. But notice the rest of the membrane is still positive on the outside, negative on the inside. So this happens only at one place, and then it's going to move on. So as that's happening, potassium over here is going to be leaking back out, And then the sodium-potassium pump is going to do the rest of the work to get us back to the resting potential. So back to resting is we need to get back to being positive outside and negative on the inside. But notice our action potential has now moved down a step over here, and so at this point the sodium has been moving in, and so we've got a switching. So I'm going to show you guys a animation of this so that you can kind of see it more in action. Okay, so in this animation we're going to have a stimulus come into the cell. So you can see the stimulus comes in, remember, the dendrites, down the cell body, and then down the axon. What this actually looks like on the axon, so here we have our axon at rest. So again, positive on the outside, negative on the inside, and that's because we've got lots of sodium outside. As the action potential comes in, that's the stimulus just came in, sodium rushes in. And that now makes us positive on the outside and negative, or positive on the inside, excuse me, and negative on the outside. Now this is going to move down. So the action potential moves down to the next step. And as that happens, our potassium is going back out so that we can get back to resting. So that step has to occur to get us back to resting. And this happens in this step-by-step process all the way down the entire neuron. Okay, so all we're doing is switching potassium and sodium to make the action potential move down, and then we have to get back to the resting potential each step along the way. What actually happens when we are talking about our neurons is our neurons are actually myelinated. That means that they are covered in these little bundles of fat along the way. We call those the myelin sheaths. They're made by Schwann cells. And then in between the myelin sheaths, you've got the nodes of Ranvier. The nodes of Ranvier are where the action potential is actually going to happen. So what this does is it allows the process to jump from one place to the next. So this kind of shows you the difference between them. So if you have your impulse coming down through your cell body and there are no myelin sheaths on the axon, so here it's going to move down and it's got to go through every single part of the axon. So after 10 milliseconds, the action potential has only gotten to this point. Whereas if you have the myelin sheaths, the action potential will actually jump over. So here it's jumped, and then it jumps again. So after 10 milliseconds, the action potential has moved much further down the neuron. And so that allows our nervous system to move and work a lot faster. And these myelin sheaths are made of fat, and they are formed mostly during our first couple of years of life, and that's why a high-fat diet is important at that time. So again, I'm going to show you what this looks like in the animation so that you can see the jumping. Okay, so if we send an impulse down this particular neuron, this neuron has the myelin sheaths all along it. So you're going to watch as the impulse is actually going to jump. So in comes the impulse, and then the action potential is here. And what it's going to do is it's going to come down and it jumps over each of these little spots, and that makes it go much faster. If it had to go along each little piece, it takes longer, as that last diagram just showed us. Okay, so again, when they can jump along, it goes in a lot faster manner, and you can get your action potential to go much further along, and the more myelin sheath you have, the more insulated it is, the faster the saltatory conduction can occur.